Good afternoon, ladies and gentlemen. Welcome to our REFLECT webinar on experiments for improving the efficiency of geothermal plants. This webinar will present methods developed and results obtained during the Horizon 2020 project REFLECT, which deals with redefining geothermal fluid properties at extreme conditions to optimize future geothermal energy extraction. My name is Katrin Kieling, and I'm working at the Helmholtz Centre Potsdam, the German Research Center for Geosciences, in the section Geoenergy. I'm the project manager of the REFLECT project, and I'm mostly responsible for all the organizational issues in the project. But REFLECT has 14 European partners, which include universities, research institutions, SME, and industry, and come from 10 European countries. The aim of REFLECT is to collect chemical, physical, and microbiological data of geothermal fluids to better understand the processes which influence these properties during the operation of geothermal power plants. Today, we concentrate on a selection of experiments which were performed for REFLECT and the results and the knowledge which we gained from these experiments. To give you a short outline of today's program, so we will start with a general presentation by Simona Regenspurg, the REFLECT coordinator from GEOTHERMAL Potsdam. This will be followed by a presentation of Serhat Tonkul from the İzmir Institute of Technology on technical problems in geothermal power plants. And after that, we hear from Chris Boeije from the Technical University of Delft about CO2 degassing kinetics in bulk and porous media. And finally, we have the presentation by Danaé Bregnard from the University of Neuchâtel on assessing the microbial diversity in deep geothermal fluids. We will then afterwards take time for questions and answers, and I encourage you to write your questions already in the Q&A tool during the presentations or after each presentation, such that we can attend to the questions after the talks. Then I have a last remark before we really start. This webinar will be recorded, and my colleagues Maria Lopez and Alberto Sanchez from the European Federation of Geologists, who are organizing this webinar, they will publish the recorded webinar on YouTube afterwards, and also the presentations of the speakers will be provided to you, to the participants after the webinar. Having said that, it's now my pleasure to introduce you to the first speaker. Simona Regenspurg works at the Helmholtz Centre Potsdam, the German Research Center for Geosciences. Simona studied geology and hydrogeology at the Ludwig-Maximilians University in Munich and completed then research stays in Sweden, the US, and Switzerland. Before then, she came to GEOTHERMAL Potsdam in 2009. In the section Geoenergy, she leads a group working on geothermal fluids, and her main research interests are geochemical processes that take place during the operation of geothermal plants. She initiated this project idea for REFLECT, and she's now the coordinator of the REFLECT project. And additionally to her research, she also teaches at the Free University in Berlin. And she will now give a short introduction of the REFLECT project. And with that, I hand over to you.
Thank you, Katrin, and have a good afternoon to all of you. Yes, so welcome again to this last webinar of our series, this time on the experimental part of the REFLECT project. REFLECT stands for Redefining Geothermal Fluid Properties at Extreme Conditions to Optimize Future Geothermal Energy Extraction. And before we come to the actual talks of today, I will give you a short introduction to the project. Sorry. Yes, so the REFLECT project answer an EU Horizon 2020 call on optimizing manufacturing and system operation. It started in January 2020. It goes for 3 years, so we are almost finished by now, and it's coordinated by GEOZ. As Katrin said, we are 14 partners from 10 countries, from research, mainly in research institute, but also industry and governmental institutions. The background of the whole REFLECT project is that Many geothermal operators have massive problems due to scaling and corrosion. You see here on the right-hand side some pictures of scaling and corrosion, so mineral precipitation, clogging the pipes and wells, and that leads to downtime of the plants, maintenance costs, and even failure of geothermal installations. And the reason why we have that is because often some properties of the geothermal fluid are not very well understood, and therefore they cannot really well predicted, and the plants are not prepared basically for these reactions. And the purpose of the REFLECT project is to better understand the geochemical and geophysical properties of the geothermal fluids. And these unknown properties I'm talking about, this is shown here in this diagram. So most geothermal fluids can be classified according to temperature and salinity. You see that in the diagram here that the fluids can have very high salinities or temperatures up to 400 gram per liter, salt content of 400 gram per 400 degrees C. And there are some types of fluids, we mark them either type A fluid or type C fluids, so the blue shaded areas. These are very typical fluid either from like sort of sedimentary basins, for example, that can have its very high salinities, but typically not so high temperatures, or we have the type A fluids with very high temperatures, more than 200 degrees, up to 400, but usually lower salinities, like up to seawater salinity. For the other types of fluids, of course, they also exist, but they are not so such a challenge, because here the properties are better understood. So for type A and type C fluid, here many properties are not well understood, and this shows the dashed areas in the diagrams. For example, the question when what about gas solubility, the orange area, when do first gas bubbles form at the extreme conditions? What about mineral solubility and fluid physics? At which temperature, for example, does scaling occur in high saline systems? For the high temperature system, the silica precipitation kinetics are very difficult to predict. And then for the lower temperature system, it is known that microbiology and organics play a role, but it's not clear until what is actually the role in this system. So these are the areas the REFLECT project wants to investigate, and our overall objective is to do, in the end, accurate predictions by thorough knowledge of the physical and chemical properties of the geothermal fluids. The concept is from react to reflect. So we want to avoid problems by reflecting first related to fluid chemistry, rather than just treat the problems. For that, we defined 7 goals in the region REFLECT project. The goals are listed on the left-hand side, and the structure of the project is shown on the right-hand side. So the structure follows the goals. So what we want to do is to extend databases for mineral precipitation to higher temperatures and higher salinities in field lab and modeling. So this is basically the work packages 1 and 2. So we divided the very high temperature fluids and the very high saline fluids and properties here. So extend databases, extend the location of the degasification front for geothermal fluids. We want to determine the types of organic matter and microorganisms in geothermal fluids. The physical properties at various pressures, temperatures, and salinities. So these 4 goals actually are all included in the data generation part of the project structure. Also to develop a downhole sampling techniques for hot and super hot systems, because without good samples, one cannot do any good analysis. And then we have 2 goals that are more related to the data application. So what we do in work package 3 is to develop a fluid atlas for Europe, and in work package 4 to do system modeling. so geothermal transport modeling, for example. But today we focus on the experimental part, and here on the three goals, project goals on mineral solubilities, degasification, and microorganisms. But for doing so, we want to understand, of course, the properties of the natural geothermal fluids. So within the project, we have a couple of geothermal sites, either high salinity sites marked in green here, high temperature sites marked in red, or some sites that are both high saline and high temperature sites. And in these sites, we did a very detailed characterization of the geothermal fluids. So we collected samples, determined, of course, chemical parameters, the most important cations and anions, gases, and looked for scales that was investigated, like the chemical composition and the mineralogy of the scales. And we looked for organic components, classified them, and for the microorganisms at these sites. And from these natural observations, we then go into the lab to do experimental characterization of geochemical processes. But here we use synthetic fluids that are similar to the real fluids, and did experiments to determine gas solubility, mineral solubility, or kinetic data. Of course, for these experiments, we want to investigate the impact of salinity, of pressure, temperature, metal impurities, or organics and microorganisms. And with that, in the end, we have the geochemical data that can extend databases for geochemical models, and can be used to do actually predictions of chemical reactions. For these experiments, that is actually the most challenging part of the project, because we need equipment, lab equipment that can go up to very high temperatures and salinity. So all our autoclaves, you see here a couple of examples from different research institutes. They are all very specific and very tricky to operate because they have to be corrosion resistant and operate at these high pressures and temperatures. And with that, I'm coming to my last slide to introduce actually our speakers from today. These are all young researchers. So we have three, the project itself, we have 3 PhD students, 8 master and 3 bachelor students integrated in the project, and 2 of the PhDs and 1 young postdoc will present today about their experimental results. And yeah, with that, I'm coming to the end of my first introductory presentation. Want to mention the webinars we have posted on YouTube that already took place. And of course, this one will also can be found on YouTube in the end. And if you want to see us live and in 3D, you can join us at the EGC in Berlin in October 2022. And yeah, of course, you can find more information on our webpage, on LinkedIn, and on Twitter. And I thank you for your attention. Thanks.
Thank you very much, Simona, for the introduction to the REFLECT project and the experiments. As I said before, we will have the question and answer session after all the talks. And please, if you have any questions, type them into the Q&A tool, and we will attend to the questions after the talks. And now we warmly welcome Serhat Tonkul from the İzmir Institute of Technology. Serhat graduated from the Çanakkale on Smart University Geological Engineering Department in 2014. He was very interested in energy resources, hydrology, and groundwater during his undergraduate, and he wanted to develop this field further himself. Then he worked as an intern engineer at Maren Maras geothermal power plant in Aydin or Germencik in 2012, and at the Turkish Petroleum Corporation in Ankara. He graduated with a master's degree from İzmir Institute of Technology, Energy engineering department, and he's currently a PhD student at İzmir Institute of Technology, the Department of Environmental Science and Engineering. He carries out his doctoral thesis studies on 3D modeling, geochemistry, and numerical modeling, and he will present today his work on the technical problems in geothermal power plants. And with that, Serhat, we are looking forward to your presentation.
Thank you so much, Katrin. Sharing the screen. Good afternoon, everyone. Again, I'm Serhat. I'm a PhD student at İzmir Institute of Technology. Thank you for joining us at this webinar. Today, I will try to give you information about the technical problems frequently encountered in geothermal power plants. I will share how we deal with these technical problems and how we minimize them. So firstly, let's talk about what geothermal energy is. As you know, geothermal energy is the energy stored in the form of heat beneath the surface of the solid earth. It's clean and sustainable energy. Resources of geothermal energy range from the shallow ground to hot water and hot rock found a few miles beneath the Earth's surface and down even deeper to the extremely high temperatures of molten rock called magma. Therefore, a geothermal system is characterized by heat source, convective fluids, and meteoric recharge, of course. So, geothermal energy has different uses in the world. Heating and cooling applications and electricity generation are a few of them. When we look at the countries that use geothermal energy for electricity generation, as you can see in this figure, Turkey, Italy, and Iceland are at the top. Also, Iceland, France, and Turkey are used geothermal energy for heating and cooling purposes. When we look at the heat distributions, when we look at the geothermal heat distribution map at 2000 meters in the world, the heat distributions vary between 100 and 200 degrees in Germany, 60 and 100 degrees in France, and 100 and 200 degrees in Turkey, especially in the west part of the Turkey. So in Poland and UK, Romania, the heat distribution is lower than in countries and is in the range of 40 and 60 degrees. And almost of these geothermal power plants have different technical problems. So let's look at these technical problems now. We can classify the difficulties encountered in geothermal power plants as before production, during production, and after production. Difficulties before production include fluid incompatibility and clay splitting. So physical problems can occur both before and production processes, and chemical processes and biological processes can occur during production and after production also. These problems can be seen in production wells, reinjection wells, transmission lines, and even surface equipment system in the geothermal power plants. So the most common technical problems are chemical problems, of course. Among these chemical problems, scaling and corrosion are the most well-known. So difficulties encountered in geothermal power plants may also occur during exploration phase, but challenges during exploration can be minimized with proper resource management, successful drilling, and collaborative working of engineers and site officials. If you look at the challenges that occur during the engineering part, these can also be minimized by the selection of appropriate surface and subsurface equipment and the use of appropriate surface equipment and the use of monitoring wells to monitor the production and reinjection wells also. Operational difficulties often occur after production. Here, the sustainability, efficiency, and safety of the geothermal power plant are important. If we can, of course, if we can increase the efficiency of the power plant, we will also ensure the sustainability of the power plant. Increasing the efficiency of the power plant depends on the solution, scaling and corrosion problems, which are the most common chemical problems in geothermal power plants. So let's look at these problems now. Firstly, let's look at the answer to what a scaling is. So as you know, the scaling is basically known as mineral precipitation. The terms scaling or scale formation are commonly used when the precipitate form is a hard deposit, of course. So pH, temperature, water chemistry, and reservoir lithology are the most important factors affecting scale formation in the power plant. So scale formation may occur almost anywhere in the geothermal power plant. It may occur geothermal formations, geothermal wells, surface facilities, reinjection systems, cooling systems, and also even turbine blades. So when we look at the scaling types in the power plant, one of them is silica scaling, of course. So silica is deposited from virtually all high temperature geothermal fluids and sometimes from some medium geothermal temperature fluids, of course. The mechanism of silica deposition is up to now neither simple nor well understood. So when we look at the calcite scaling, as you know, almost all geothermal fluids contain significant quantities of dissolved carbon dioxide. So the flashing of the vapor phase and the carbon dioxide release cause a pH increase. As a result, supersaturation conditions are established and calcium carbonate is deposited. So when we look at the sulfide scaling in the power plants, in sulfide scaling, as the temperature decreases, the solubility of any sulfide scaling decreases also. The most common types of scale are pyrite, stibnite, chalcocite, sphalerite, and also galena. So, of course, these silica and calcite scaling are the most common type of challenges in the power plants. On the other hand, the sulfide scaling is rare in many geothermal power plants. So let's look at another technical problems, which is corrosion. Corrosion generally occurs after scaling in geothermal power plants because chemical inhibitors used to prevent scale formation, pH adjustment, and temperature factors cause corrosion in geothermal power plants and surface equipment. So corrosion, in a simple way, destruction of a material by chemical or electrochemical action of the surrounding environment. Of course, pH, temperature, oxygen concentration, fluid velocity, and suspended solids are the governing parameters for the corrosion problem. So in the power plants, the most common corrosion types are general and pitting corrosion. So general corrosion is defined as the uniform loss of metal from the entire exposed surface of the metal, and it occurs in geothermal power plants with an increase in the corrosion rate in the presence of oxygen and hydrogen sulfur in the geothermal fluid, of course. The pitting corrosion causes small holes on metal surfaces, as you can see in this figure. For example, the pitting corrosion causes small holes at the wellhead system in the power plant. So how do we deal with these technical problems in the geothermal power plants? For this, we carried out the field and laboratory studies, of course. We can divide these experimental studies into two as field and laboratory studies. The field studies include the collection of water sampling, scale sampling, and rock sampling also. We need to evaluate these samples taken from the field in a laboratory condition. A determination of physical and chemical parameters on water samples, isotope evaluation, and determination of mineral saturation constitute the most important part, of course. So we try to understand the elemental composition of the rock and scale samples, of course. By performing XRD and XRF and SEM analysis. So at this point, we try to find the answer to which rock types in the reservoir are related to the scale sample. So after doing these evaluations, we first have to reveal the 3D conceptual model of the geothermal system based on the structural data and well logs. We need to support this 3D model with the help of rock and scale sample analysis to better understand the reservoir lithology. We also need to understand the fault mechanism controlling the 3D thermal model of the site with the temperature data, because this temperature data, this temperature model helps us to better understand the solubility of minerals in reservoir rocks, of course. Finally, we need to verify our findings using some computer programs. The most critical factors are determining the reinjection temperature, and revealing the mineral saturations depends on the temperature and pH, of course. As you know, mineral saturation diagrams are diagrams used to show the scale types and possible scale types that may occur in the geothermal power plants and surface equipment system. So thank you very much for listening, and I would like to thank Dr. Taygun Uzelli for helping me during these field studies. Thank you so much again.
Thank you very much, Serhat, for this very nice presentation of technical problems in geothermal power plants. Again, I ask you to type your questions into the Q&A tool, so we will ask them after the talks in the discussion. And I will now switch over to our next speaker, to Chris Boeije. Chris Boeije is a postdoctoral researcher with a background in various topics regarding the multi-phase flow in porous media. He has a PhD from Delft University of Technology, for which he studied foam flow for enhancing oil recovery. During his research stays then at the Bordeaux INP and the University of Lyon, he gathered further research experience, focusing on groundwater aquifer remediation. He is currently working on a project related to degassing of geothermal fluids, again at the Delft University of Technology. And this will also be what he presents today in his talk. So it is CO2 degassing, kinetics, and bulk and porous media. And with that, I hand over to Chris. The floor is yours.
Okay, thank you, Katrin. Let me start by sharing my screen. Thank you for your kind introduction. Yeah, so I'll be talking today about the work we've been doing on the CO2 degassing kinetics in bulk and porous media. Here at Delft University of Technology, we're doing the experiments, and we're getting some help from our colleague at Geosciences on the modeling part of this. So let me start with a brief introduction and problem statement of what we're dealing with. So we're looking at the degassing of geothermal fluids. Geothermal fluids typically contain significant quantities of dissolved gases such as CO2 and methane. And as these waters are being produced, the pressure decreases towards the production well, and this can cause the dissolved gas to come out of solutions and form free gas bubbles either in or near the production well. And this can give several problems. So first of all, these are greenhouse gases. They can cause corrosion of facilities, and finally they can block the pores of the reservoir. And as the gas takes up space in the reservoir, this means that they just limit the ability for the water to flow, and thus the water production can decrease. And this is assumed to have happened at the Groß-Schönebeck reservoir in Germany. So how do we go about studying this? That brings us to my research question and our approach. So we want to look at the conditions under which these free gas bubbles form either in a geothermal well or a reservoir. And we're doing this using two sets of experiments. First of all, we have a set of degassing experiments in a visual cell where we depressurize water that is saturated with CO2. I should mention that in this presentation, I will mostly be talking about CO2 degassing, as it's one of the more common dissolved gases. And I'll be comparing the results from these experiments with a geochemical model for CO2 solubility in saline fluid that was made using Freak-C. And like I said, we have another set of experiments where we are assessing the degassing process in porous media using coreflood experiments, and where we use pressure monitoring to assess the degassing process. But let's start with the visual cell experiments. So this is kind of what our setup looks like. We have a visual cell over here on the left, to which we point a high-speed camera. The visual cell is filled with water that is saturated with CO2 and then depressurized. And during that depressurization, we are recording images of what that looks like. So at some point, you will see bubbles forming. So you get these kind of images, which you can then process to determine how many bubbles are formed. And in the meantime, you also can simultaneously record the pressure, so you can have a number of bubbles versus pressure. But here today I'll be talking mostly about the occurrence of the first bubble and how that compares to our geochemical model. So we have two sets of experiments here with two different brine salinities. Our first experiment is using just a sodium chloride solution, where we are doing experiments at 0.2 and 0.5 mole per kilogram of water of CO2 concentrations at temperature up to 150 degrees. And this is what our geochemical model for the CO2 solubility is predicting in terms of the degassing pressure. So I've here, these are contours of iso solubility in mole per kilograms of water, and I have highlighted the 0.2 and 0.5 mole contour, and this is based on an equation of state from Duan and Sun. And if I then plot my points for which where I'm expecting my first free gas CO2 bubble, I find that for our higher concentrations, 0.5 mole per kilogram of water, we can get a fairly accurate match with this model. But for the lower concentration, we are getting a much, the fit is much worse. And one thing of note is that some of these, wait, let me see if I can just put, can this still work? No. So one thing of note is that for some of these experiments, we had bubbles forming in gaseous, in their gaseous phase. So for instance, this is what we get for our 40 degrees experiments. And this is essentially what you get when you open a bottle of sparkling water. But at higher temperatures, you already get bubbles forming at much higher pressure, and that while they are still in a supercritical phase. And we can clearly see a distinct difference in terms of the structure of these bubbles. So the kinetics are much different between the gaseous and supercritical bubbles. And we also did the same experiments for high salinity brines. So in this case, we chose a lower CO2 concentrations. And so far, we've only done very low temperature experiments up to 80 degrees Celsius. So here we would expect bubbles. So we're using, in this case, 0.15 mole per kilogram. So we would expect them already at these pressures, but instead what we're finding is that they are occurring much later. So at much lower pressures, we are finding our first bubbles. And then to briefly talk about our other set of experiments, so the coreflood experiments. So this is a brief schematic of what these experimental setup looks like. So we have a Bentheimer core. Let me put my laser pointer on again. So we have either a Bentheimer core or a Berea core, and so it's high or low permeability. And in this, we co-inject either CO2 with an aqueous phase, with either tap water or a high salinity brine, and they mix already in the tubing before entering this core. And then we are monitoring the onset of degassing using pressure transducers. So depending on what pressure we're finding, we can see that the degassing has taken place. And in this case, we're using pressure up to 90 bar, temperature range, in this case 30 to 90 degrees Celsius, and CO2 concentration of 0.2 to 1.3 mole per liter. And this is the typical kind of result that we're getting from this, in this case for a Bentheimer sandstone at 30 degrees Celsius. So what we're finding is that we are doing an experiment where we are decreasing pressure inside the core. So we started at a certain pressure and then decreased the pressure. And at a certain point, we find that the steady state pressure drop in the last section of the core starts to rise. And the point at which that happens is what we find is our onset of degassing. So that is where the flow is no longer single phase, but starts to form bubbles inside the core. So here I'm plotting for different CO2 concentrations here in mole per liter. I can also express that in terms of the effective permeability of the medium to see to which extent it is being blocked using Darcy's law. So essentially for higher pressure drop means lower effective permeability. Then to see whether this onset of degassing that we find here kind of agrees with theory, we can compare with the with solubility theory. In this case, we're using a fairly basic theory called the van 't Hoff equation, which is essentially a temperature corrected version of Henry's law. And we can find that this fairly accurate estimation of when we can expect our first degassing. And like I said, we did this for two different rocks, also for Berea sandstone, which has lower permeability and also smaller pores. And as a result of this, we find that in this case, you find a much more abrupt change between a state of single phase flow and multi-phase flow there. So in this case, yeah, the change between, well, the pores are blocked much more quickly than for the Bentheimer sandstone. So here at just pressure slightly below the degassing pressure, you already get a reduction of the effective permeability of up to 90%, whereas for the Bentheimer you only find up to 50% reduction. But both the Bentheimer and the Berea, they do agree in terms with the van 't Hoff equation in terms of when the first degassing is found. Then we also briefly look at other things like the effect of temperature and the effect of salinity. So for temperature, we find in this case, well, I'm plotting here two different experiments, two different saturations. We find that at higher temperature, you find some variation from this theory. But we are finding that higher temperatures also mean higher bubble point pressures. And the same goes for higher salinity brines. You already find increased pressures at which the degassing takes place. And so we did that for three different concentrations. We find that for higher concentration, we find a larger shift in when we can expect the first degassing. So where do we go from here with these experiments? So currently we're looking at doing these experiments also inside a CT scanner. So we can get data on the water saturations and gas saturation inside that rock to see how that how the bubbles are formed there. So that brings me to our conclusions. So for the visual cell, we found decent agreement for the higher concentrations with the Duan and Sun equation of state model, but not so much for the lower concentrations. And for the high salinity experiment, we find even higher deviations from the model. And in terms of comparison with the van 't Hoff equation that we found for the core floods, we found for both the Bentheimer and Berea rock cores, we found good agreement at moderate pressure and temperature conditions, and found a much more rapid decrease of the effective permeability for the Berea sandstone compared to the Bentheimer. But also here we found some deviations from the theory at higher temperature and salinity. And that was all I have for you today. So thank you for your attention. If you have any questions, feel free to post them in the chat. Thank you.
Thanks a lot, Chris. As Chris has already said, post your questions in the Q&A tool so that we can ask them afterwards. And I will directly continue now with the presentation of our last speaker, with Danaé Bregnard. After a bachelor in biology at the University of Neuchâtel in Switzerland, Danaé Bregnard continued with a master's thesis in microbiology to obtain the master in biology at the same university. This work was focused on fungal endobacteria, which compose a large part of the microbiome of fungi. Danaé is currently pursuing a PhD degree in microbiology at the University of Neuchâtel, and this time with a focus on microbial diversity present in deep geothermal fluid. This means, in addition to fungi, she now also looks at bacteria and archaea. Danaé wants to assess the microbial diversity present in deep geothermal reservoirs, as well as she's looking at which of these microorganisms are able to grow inside the power plant system. And with that, Danaé, I'm happy to welcome you today, and I'm curious to learn more about your work.
Thank you for the nice introduction, Katrin. So I will share the screen. So as Katrin mentioned, today I will talk about the microbial diversity that we can find in deep geothermal fluids. And if you think about the Earth and all the ecosystems it contains, I think you will agree with me that some of these ecosystems are extreme for life. And even though they are extreme, such as hot springs or underwater volcanoes or salt flats, for example, these ecosystems can still have some microbial life in them. So as Katrin mentioned before, we will focus on the microbial life composed of bacteria, archaea, and fungi. Bacteria and archaea, they are the most extremophilic microbes, but fungi can still be present under extreme conditions. And if we look at all these extreme ecosystems, as you can see here, we have detected in the last few decades some microbes in these systems. And if you look at geothermal reservoirs more precisely, then bacteria and archaea have been already detected in some of these systems. However, fungi were not detected yet, but it doesn't mean that they are not present, but maybe it means that we didn't look at fungi in these extreme environments, because the studies on the microbial diversity in these systems are still scarce. So that's why within REFLECT, we had 8 sampling sites all around Europe. So some are geographically close to each other, so that's why you don't see all of them on the map. And with all of these different sampling sites, we had fluids with a temperature that ranged from 47 degrees to 345 degrees. And there was also highly saline fluids and a variability in the pH or in iron content, etc. So having all of these diverse samples will allow us to have a broader view of what happens in terms of microbes in geothermal fluids in Europe. If we focus on the microbes, so on bacteria, archaea, and fungi, one thing that they have in common, like all living organisms, is that they all contain DNA. And this is highly useful for us because we can look at all the DNA that is present in the samples. And with that, we will be able to know which type of microorganisms are present. So this is what we do in part of the project. So we use all these molecular methods to look at which microorganisms are present. But on the other hand, we also want to use culture enrichment methods. And so we will do liquid and solid cultures to try to isolate these microorganisms and to cultivate them in the labs. And that will allow us to characterize them further. But when we do these culture methods, one thing to keep in mind is that many microbes coming from the environment will not be able to grow in the lab. And this is specifically true for microorganisms coming from extreme environments. Because if we don't really know the microorganisms, or we don't really know the conditions under which they grow, then it's difficult also for us to give them what they need to grow in the lab. So now I will show you some of the results we have for both of these methods. And I will start with the cultures. So as you can see on the pictures, we managed within REFLECT to isolate both bacteria and fungi. And so they all grow in the lab and they are still under characterization, but I will present them shortly to you. So first of all, we have that Thermaerobacter bacteria, which is, as you can see on both pictures, a filamentous bacteria. And this bacteria was isolated at 60 degrees, but it can grow from 40 degrees to above 85 degrees Celsius. So we didn't reach the upper limit of the temperature for that bacteria yet. Then we also isolated that Bacillus bacteria. So the genus Bacillus is highly common in different types of environments, also in environments that are not extreme. Here we don't know the specific species of the bacteria, but we know that it's genetically related only to extremophilic Bacillus strains. And as you can see on the picture on the right, so it's able to form these white dots. And actually these dots are bacterial spores. So these spores are resistant structures that allow the bacteria to go into a form of dormancy. So it allows it to survive some extreme conditions and then to start growing afterwards when the conditions are suitable for growth. And so this bacteria is able to form some spores. It's also the case for the bacterium I presented to you before. And then on the fungal side, we have that Penicillium citrinum. And as you can see also in both pictures, it is able to form spores. It's these round structures that you can see on the pictures. And so these spores also allow the fungus to survive harsh conditions. So fungi, they are less tolerant to high temperature compared to bacteria or archaea. And so this one is only able to grow up to 35 degrees Celsius, but it's already high for fungus. And as I mentioned before, it's able to make these spores, so it's able to resist to higher temperatures. One thing also that is interesting with that fungus is that we know that the other strains of that one are able to produce some pigments with varied properties. And we tested that on our fungus. And as you can see on the picture on the right, it is able to produce some pigments. So in the huge bottle, you have the initial growing medium that is brown. And in the falcon tube, you have how the medium looks like after the fungus grew in there. So it's able to produce a lot of yellow pigments. And what the fungus is producing is still also under characterization. So that was a quick overview of the culture enrichment methods that we did. And now I will shortly introduce you to the molecular methods. So when we look at the DNA that is present in the samples, then we end up with something that looks like that. So here it's only an example with a few samples. And on the graph, you have each sample, and then each color represents one bacterial species. And that graph you can here split it into two. On the left, you have the spores, because we did a special treatment on the samples to be able to get the DNA from resistant cells, just as the spores. And on the right, we only did a direct DNA extraction to get the DNA from all other types of cells. And what you can see here is that obviously there is some bacterial diversity present in the fluids. So this is the first take-home message. And then the second one is that if you look at the production site here, it's interesting to see that there is less diversity at the production site compared to the injection site. This is not so surprising for us because when you bring up the fluid, then the conditions are extreme. And then you can take the heat from the fluids. And after that, the microorganisms have other conditions and maybe they are able to develop in the pipes. And that's maybe why here in this case, there is a higher diversity that is detectable at the injection site. So with that, I hope that you will agree with me that there is a diversity of microorganisms that can be present in geothermal fluids, that we should further investigate that. And that's what we do within REFLECT. And with that, I'd like to thank the whole REFLECT team, my lab team, for their support, and I thank you for your attention.
Thank you very much, Danaé, for this very interesting presentation on the microbial diversity. We will now start the Q&A session, and we have one first question, which I think is more or less directed at Simona. What about interactions between different types of geothermal fluids and pipe-made materials? Which pipe materials avoid maintenance, high costs, and time?
Oh, that's a very difficult questions to answer. Difficult on the one hand side because many factors can be responsible for the for corrosion. Many factors in the fluid, and on the other hand, corrosion can always be avoided by using highly alloyed steels, but that is a question of cost. And usually most geothermal plants use simple carbon steel for the casing and for many other parts, but they are prone to corrosion. So it is, one always have to consider what is my financial situation, how can I avoid certain types of corrosion at least, and then how much is the are the costs in the end. I mean, first of all, you have to know what are the properties that cause the corrosion. Is it salinity? Is it H2S? Is it a CO2? Just acid pH? So that many factors can cause it. And then question is, do I use titanium or hastelloy steel or simple carbon steel and just exchange it once in a while? So it is difficult to answer. I think many operators struggle here. And but it's good to know beforehand what are your properties of the fluid in order to find the best material. So, but there's no simple answer. I'm sorry.
Thanks a lot, Simona. We have another question. According to the geothermal gradient in Turkey, which are the main scales precipitated? I think that's a question for Serhat.
Sorry. Actually, in Turkey, we can see different types of scale in many geothermal power sites. Because, for example, in western part of Turkey, in Aydın region, we can see a stibnite scaling because of the reservoir lithology is different from any other site. For example, in the Tuzla region, we can see silica-based scaling. The silica types encountered in the geothermal power plants actually depends on the reservoir lithology and the geology in this site. Actually, there is no specific scale types in Turkey. Actually, we can say different types of scale in Turkey.
Okay, thanks a lot. I have a third question, which is directed to Danaé. How are fungi and bacteria coping with high pressures? What is the maximum pressure that they can survive? On the pressure.
So for the pressure, it depends if they are actively growing or not. So if they're able to form these types of spores, as I've shown you, these spores are highly resistant to pressure because they have a hard cell envelope. So if they are under this dormant state, then they easily can deal with pressure. But by heart, I don't remember the level of pressure they can survive.
Okay, and actually I have a follow-up question to this one. You said that the fungi, they also form spores, but they themselves cannot survive high temperatures. But what's the level of temperature for the spores for fungi?
So for fungi, I'm not sure if it is known the temperature that they can survive under the form of spores. So it has been recently discovered that they can survive high temperature, like in hot springs, but I think it's still unknown which is the upper limit.
Okay, thanks. And there was also another question to you. Stay tuned. It was, can we grow penicillin if it can be used for medicine, then industrially in thermal water?
So it's true that the penicillium fungi can be used in medicine if they produce that penicillin, but there's a lot of different species of penicillium. In our case, then we will see with the characterization of the compounds if it's produced something like that or not. And if yes, I guess yes, we can grow them in geothermal fluids like the one they've been isolated from. But we will still need to give them good condition for growth.
Okay, thanks. I have a question. Apart from corrosion, what other main problems are caused by microorganisms? Oh, that's also to you, Danaé.
I'm sorry, can you repeat your
question? It was apart from corrosion, what other main problems are caused by microorganisms?
So they can either induce mineral precipitation, and they can also, depending on the settings, block the filters because they can form these slimy biofilms. And then they can, if they're able to grow, they can nearly block everything in the pipes.
Thanks a lot. Then I have another question to Serhat. Concerning the impressive growth of geothermal development in Turkey in the last years, are there more geothermal projects in the close future in Turkey?
Yes, of course. There are many projects in Turkey because in western part of the Turkey is developing in terms of geothermal projects. There are many springs waiting to be discovered right now, and we can say that there are lots of geothermal projects, especially in Western Anatolia in Turkey.
Okay, thanks. Then I have a question from someone who says that he's not in the geothermal energy field, but was asking for, out of curiosity, is the induced fluid chemistry aligned to the fluid chemistry existing in the wells? I'm not sure what the question is. Maybe Simona, you want?
Well, try. I guess it's referred to a well doublet where you have one well where the produced fluid comes out and the other usually the same fluid, of course, where you just inject. So you take out the heat from the produced fluid and re-inject the cold fluid, and then the chemistry changes a little bit due to chemical reactions that happen. For example, if you have precipitation of certain minerals and these elements are removed from the brine. And sometimes the operators add also chemistry, like chemical components to avoid the scaling, like inhibitors, corrosion inhibitors, scaling inhibitors. So these components also, of course, enter, or oxygen could enter the fluid. So the chemistry changes indeed a little bit sometimes.
Okay, thanks a lot. I have a question to Chris. The supercritical bubbles, they seem to be much smaller than the not supercritical ones. Is it correct? And what is the implication for geothermal operation?
Yes, it seems to be correct. For what are the implications is that well, first of all, these only seem to occur so far that we could see at very high concentration. So I'm really not entirely sure if that it really happens often in the field, because normally you would have perhaps slightly lower concentration. But yeah, we do think it is feasible that there are conditions where that happens. Yeah, we would say that bigger bubbles tend to be lead to increased blocking of pores. Like, well, the bigger the bubbles, the easier it is to block pores. So probably for these supercritical bubbles, they will be a bit less in terms of blocking the pores. That would be my, well, initial guess at least.
Yeah, to me, it also seems that they are moving faster. I mean, maybe that's also artificial, but this would also indicate that they somehow move faster through the reservoir then, maybe.
Yeah, well, in terms of moving faster, I'm not so sure about that. I think what we saw in our experiments that they have, yeah, they seem to just all form at once at a single temperature, but in terms of movement, they may be not, in fact, not quite as fast initially because of, yeah, just reduced buoyancy in our cell at least. But yeah, we did see some effects, like we have maybe some movement coming from like if bubbles form on the walls or so, that which might be hotter than the surrounding fluid, then you can have maybe some effects from that you have some yeah, movement might be impacted by just temperature gradient.
Thanks. And I also have a follow-up question to you. You said that for the experiments which is did with the rocks, there are deviations when you have different, or when you increase temperature or salinity. So do you think that more experiments are needed with different temperatures and with salinities to understand the process even better?
Yes, and also I think there we could just use maybe perhaps a more sophisticated model to capture that, like we did already for the well, at least what we've been trying to do for the visual cell experiments, like this freak-C coupled model. Whereas for the yeah, porous media experiment so far, we just compared with a very basic Henry's law based model. So yeah, we would need some additional analysis perhaps to see if we can get a better fit there.
Okay, great, thanks a lot. I have another question to Danaé about the last figure you showed. In the production, there was only one species which formed spores, but many species, or some species which you extracted from the DNA. Does this mean that only this one species forms spores, or was it that you cannot detect the others?
So the others can also maybe form spores, but it means that the resistance of the cells or the resistance of the spores is slightly different. So that's why you can detect with one method or the other. Okay. So this allowed us to have all types of cell in the analysis.
Okay, great. Thanks a lot. I think I have no more questions. So if anyone has an urgent question, please type it now. No. So if there are no more urgent questions, then I thank everyone for attending the webinar. As I said in the beginning, the webinar was recorded. You can re-watch it on YouTube. It will be published in the next days. And also we will send around the presentations to the participants. And well, I at least enjoyed the webinar. And thank again all our speakers. Maybe you can turn on your cameras once more, then we can all see you. And with that, I wish you a very nice afternoon, a nice evening, and we see each other again. Well, maybe at the final conference at the EGC in Berlin, or even at the stakeholder workshop, which we will have already next week in Izmir. Have a good afternoon. See you soon. Bye-bye.
Bye-bye.
Bye.
Bye.
Bye, everyone.